Hello everyone, my name is Erin Francispillai, and for my project, I looked into the relative influence of abiotic and biotic predictors of specialist bee distributions. This was supervised by Dr. Jessica Forrest and Dr. Alana Noyokaitis-Lewis. Species distribution models are great tools to connect species observations to environmental variables and provide a way to estimate a species' geographic range. Traditionally, only climate variables have been used to represent the environment, but more recently, studies have shown that biotic interactions can significantly improve model performance. Among the studies that have been done, few look at dietary specialists, specifically within pollinators. And so to study this, I looked at Andrena eriginae and Andrena violae, as they both depend on one plant genus, either Claytonia or Viola. These bee species are mainly in the eastern United States and southeastern Canada, but their host plants are present throughout both countries. And so I wanted to look into what factors may be restricting them, comparing climate variables to biotic interactions, such as their host plant. So in order to represent the host plant, I'd use their predicted habitat suitability, as I wanted to see if the incorporation of the host plant's habitat suitability would help explain the restricted distributions of these bees, particularly in northern regions such as Canada. So first I needed to model the host plant's habitat suitability in order to have this variable ready. For the environmental variables, I'd picked 6 bioclimatic and 6 monthly climate variables from historical records, and using the host plant's occurrence records, here Claytonia, its distribution was modeled using a maximum entropy modeling software called Maxent, which gave me Claytonia's predicted habitat suitability. And so the same process was done for the Andrena to get its predicted habitat suitability, but now I could compare these results to another species distribution model made with the inclusion of its host plant, Claytonia. And so I'd have one model with climate variables called climate and another model with climate variables and its host plant called climate plus plant. This whole process was done for Andrena violae as well. Okay, so after running Maxent, the outputs were compared using their area under the curve, or AUC. This discriminates between the areas of high and low probability of the Andrena occurring there. In other words, values closer to 1 would mean the variables chosen were strong predictors of the bee's range. And as you can see, between the models, the values were pretty similar, which ultimately means that including the host plant didn't really add much to the model. And so I also looked into how important each individual variable was, which was measured by its permutation importance. And so all these variables at the bottom are the different climate variables, and the one at the end is the host plant. For Andrena eriginae, its host plant had contributed a lot. It was very important relative to the climate variables. These results were projected all over the United States and Canada, and you can see that with the inclusion of the host plant, there were actually fewer areas that were suitable for the Andrena. These maps were done for the host plants as well. These same regions were not too suitable for the Claytonia, and considering its importance, it would make sense that these regions are also not suitable for the bee. Um, you may notice that there are some areas here of higher suitability, but like why aren't the engineer there now? It's possible specific species within the host plant genera are more important than others, and they would have more impact in determining the andrena's range. In terms of the northern regions, it may be linked to climate, as colder temperatures have seen to affect the flowering time and thus reducing pollination probabilities. Finally, other interactions like predation or competition, also soil quality, may also be playing a role, but they were not examined here. So the same process was done for Andrena violae and its host plant. The area under the curve, again, had very similar values between the models. The host plant did contribute, although climate continued to have a higher importance. Again, into the projections, there was actually more suitability predicted when the host plant was included. But again, this can correspond to areas of higher suitability from the host plant from these green pockets seen here. Overall, yes, biotic interactions do contribute, but in this case, it was not enough to change the model's overall predictive power. There may also be specific host plants that are more important for the bee's range, and cold temperatures could be influencing the northern range limits. Finally, there may be effects of the study's scale. Previous literature has supported the idea of climate governing more large-scale distribution patterns, while biotic interactions are much more small-scale and region-specific. Overall, climate is a good predictor of the broad geographic ranges, but biotic interactions such as the species host plant can have a strong effect and may be decisive in facilitating proper range shifts. Thank you.